Forward. Right. Okay. Well, welcome, members and friends of the Royal Society of Chemistry, to this evening's lecture. I'd like to take this opportunity of not just welcoming all of you who are here in Belgium, but some of you who are abroad and who are able via these virtual meetings to join us virtually. We've just been discussing with Tim and Fabio, and I think what we will try to do in future when we get back to having meetings in person is to record them and put them on the website so that even our members who are abroad will be able to keep in touch with us in that manner. This evening, I'd just like to remind you that when we start the lecture, if you could switch off your cameras and your microphones, that would be very helpful. We don't necessarily want to see your background, particularly if you happen to be having your dinner or an early or a late aperitif. And everybody at the end can ask questions, even the younger persons. So don't be afraid. Just stick up your hand and we'll ask you to give your question in. So this evening we're very pleased to have another visit from Nick Lane, who is a professor of evolutionary biochemistry at University College London. He's actually a former Imperial chemist, Imperial College graduate. I suppose you must be from the date of the time when the theme song of students and staff at Imperial was "Workers of Imperial Unite." You've nothing to do. This is an in-joke between the two of us, because in those days the penthouse apartment on the top of the biochemistry department was occupied by Sir Ernest Chain, the professor of biochemistry, and his beloved wife. And he was a bit of a dictator anyway. Leaving that aside, you remember those days. I thought I was getting it just about right. Nick has been involved in studying metabolism and genetics for a long time, and evolution in particular. And he's written 4 books to date, which have won awards. The first one in 2002, which is dear to my heart, was Oxygen, the Molecule That Made the World. Dear to my heart, because we, as inorganic biochemists, we know just how important oxygen is in all sorts of reasons in life processes. Then we had the one which I still think is the absolute winner, Power, Sex, Suicide, and the Meaning of Life. I don't think we can beat that target with anything that you come up with subsequently, but that's a fantastic target. Then we had Life Ascending, the 10 Great Inventions of Evolution. And the most recent one you've written is The Vital Question: Why is life the way it is? So now what you're going to talk to us about this evening is, however, how does chemistry come alive, which is a sort of going back into the dark ages and seeing what happens when you try and mix a few things together. In event, yes. So hydrothermal vents and the way hydrogen and carbon dioxide might react. We're looking forward very much to your talk, Nick. Thank you very much for joining us. How does chemistry come alive? Over to you, sir.
Thank you very much. Let me see if I can share my screen first of all. Can you all see that?
Yep.
Excellent. Okay, so, well, thank you very much for the second invitation here. I did talk about the origin of life last time. And actually, most of my work is on that now. Most of my research is on that these days. And I thought it would be nice to give you an update in case a few of you remember the last time. It was probably about 5 or 6 years ago now, so quite a lot has happened. And the title, I like to play with words, as you can imagine from trying to write books. And this is about as close to the real heart of the nub of the question as I've been able to get, because the real issue, I think, for a lot of people in the field, this word alive has no meaning to most people and different meanings to those who worry about it. But that is essentially how does inorganic chemistry and organic chemistry eventually give rise to something that we would recognize as being alive? And so that's what I'm going to try and get across in the next 40 minutes or so. So as soon as you ask this question, you run into this problem about what is life. And it was probably most enjoyably addressed by Schrödinger in a famous book from 1944 entitled What is Life? And to put it in a nutshell, there are basically two aspects that he talks about. One of them is genes, and in this sense, he was quite modern in his terminology and usage. I believe this was the first use of the word code script in biology. Of course, this was during the Second World War, and encryption was very much in the air. But he'd written that chromosomes contain in some kind of code script the entire pattern of the individual's future development and of its functioning in the mature state. And that's really language that we would probably still use today in biology. But on entropy, this feels to me to be quite old-fashioned in the way he approaches it. He talks about life feeding on negative entropy, the device by which an organism maintains itself stationary at a fairly high level of orderliness really consists in continually sucking orderliness from its environment. And I have to say, I don't have a very clear vision of what he actually meant by sucking orderliness. I don't know what he had in his mind. He did have a footnote, though, where he said if he'd been catering for physicists alone, he would have let the discussion turn on free energy instead. Which is the framing that most biologists, at least, are rather more happy with, perhaps chemists less so. So before I get going properly, I would like to orient you to how I have been thinking about the question. And this is probably the easiest way of getting it across at the front, is just a few kind of key bullet points. So free energy really, to my mind, is powering growth in biology, and obviously all growth is thermodynamically favored overall. There is some kind of a reaction in the environment driving all the processes of life. So there's nothing which is truly endogonic in life. It's simply set up in a way which allows reactions to happen, and biology is in effect lowering the kinetic barriers to thermodynamically favored reactions. I'm talking to chemists, so I trust that you're all happy with that. The unfortunate thing about these online talks is that I can't see any of you, so I've got no idea if you're all kind of rolling your eyes at me at this point or what you're doing. So I'm just going to keep going, and feel free to ask questions as I'm going along, but I may not be able to answer them until the end. Okay, so heredity is a form of growth from that point of view. So the first point, growth is thermodynamically favored. Heredity is a form of growth. It's doubling by exact copying. And so all forms of selection, of natural selection, not just genetic selection, but there are other forms as well, depend ultimately on growth. And so from my point of view, at least, growth comes first. There's got to be some way of making more of yourself, and that has to happen before heredity begins. And then growth combines carbon and energy metabolism. Energy metabolism is basically coupled to the endogonic aspects of growth, but the whole thing is exogonic overall. It's really the environment itself which is driving it. And that's a key point, I think. It is, again, a gross generalization, but biochemistry as a whole, especially the core metabolic biochemistry, is pretty close to equilibrium. And growth then is driven by environmental disequilibria. Okay, another slide of bullet points, and then I'm into the talk proper. So growth starts in biology. Growth is starting with CO2 and hydrogen gas. That hydrogen gas doesn't necessarily have to just bubble out of the ground. It may be that it's coming as hydrogen equivalents from water, for example, from hydrogen sulfide, wherever it may be. But the simplest form is hydrogen itself. So CO2 is a dominant atmospheric gas on wet, rocky planets. It's been detected in the atmosphere of exoplanets and so on. It's known in the atmosphere of most of the planets and a lot of the moons in the solar system. And of course, to convert CO2 into organic molecules requires hydrogen equivalents. And most autotrophic metabolism in biology, not just photosynthesis, but all kinds of growth, autotrophic growth from rocks, from gases and so on, without necessarily using the sun, begins with hydrogen and CO2. Now, there are only 6 known pathways of CO2 fixation across all of biology. The one that most people would be familiar with is the Calvin-Benson cycle, which is used by plants and cyanobacteria, and that's basically involving sugar chemistry. But the other 5 pathways are basically involving carboxylic acid chemistry, Krebs cycle intermediates, one of which is the reverse Krebs cycle itself. And so to generalize this then, growth is the continuous conversion of hydrogen and CO2 in, whoops, sorry, something went wrong then. Can you still see my, I've lost my screen. Beg your pardon, but I seem to have lost all my slides somehow. I don't know where they've gone.
We've still got the growth starts with CO2 and H2O slide on the screen at the moment.
Okay, I can't see it anymore, unfortunately. I don't know where it's gone. Let me find it. Okay, I found it again. Right, good.
Good. Okay.
Thank you. Sorry about that. So growth is the continuous conversion then of hydrogen and CO2 into organics. And it's an environmental disequilibrium of hydrogen and CO2, which is driving the synthesis of carboxylic acids. So this is really trying to orientate you to where I'm coming from. These are more philosophical thoughts about how life is organized at the deepest level, and that will help to orient us to see what kind of experiments we can do from a more chemistry point of view on this. So this idea that biochemistry is growing out of hydrogen and CO2, this is the core of biochemistry that we're looking at here. So we're starting off with carbon dioxide. Just a couple of things I want you to notice. Hydrogen, I've put it in brown here. You can see hydrogen continuously, and I put CO2 in green. And so we're reducing CO2 to a formyl group, a methylene group, a methyl group, and then reacting with carbon monoxide to form acetal-CoA, or a simpler equivalent to acetal-CoA. So we now have a condensation to a 2 carbon form. More CO2 and hydrogen through to pyruvate, so we're into Krebs cycle intermediates here. Another CO2 through to oxaloacetate, through to alpha-ketoglutarate. It's CO2 and hydrogen everywhere you look. It's really quite remarkable in my mind. You'll see here as well that I've put some things in red boxes. Now, the things in red boxes, glycine, ribose 5-phosphate, glutamine, aspartate, acetal phosphate, or ATP actually in modern biochemistry. But these are the precursors for nucleotide synthesis. So if you're going to make DNA or RNA in biology, this is where you're starting. And you don't have to go very far, you can see, from CO2 and hydrogen through these intermediates to get to the precursors required for nucleotide synthesis, but you still have to get through them. This is the standard pathway. So to be able to make nucleotides under these kind of conditions, you're going to need to make these intermediates first. And so really the question is, at the origin of life, before there were any enzymes or genes or any evolution as we know it, there must have been a high flux from hydrogen and CO2 to support the continuous doubling of organic matter, whether that be RNA in the end or protocells or whatever it might have been, simply organic matter. So how was it focused into growth? And again, we can begin to get at this through thinking about the roots of the tree of life. And this is a kind of philosophical tree of life almost from Bill Martin. This is a conception more than the reality necessarily. But what you can see here, I hope you can see my cursor, we are eukaryotes. We have our genes in the nucleus, and we are composed really of a chimera of two different types of prokaryote, archaea and bacteria. The bacteria went on to become the mitochondria inside our cells, and the archaea were the host cell. And this, when Bill first put that forward more than 20 years ago now, it was a radical conception. This was really on the edge of the serious, I would say. I mean, it was, I think very few people believed him. Since then, phylogenetics, with enormous numbers of sequences of bacteria and archaea and eukaryotes, have shown that this is basically the case, that the eukaryotes are a derived domain, and the common ancestor of all of life was the common ancestor of the bacteria and the archaea. And you can see that Bill has got them coming out of what looks a lot like a hydrothermal vent down at the bottom here. Luca is the last universal common ancestor of all of life. And so Luca was the ancestor of the bacteria and the archaea, and potentially lived in hydrothermal vents. I will try to explain why he put that idea forward and the work we've been doing based on that idea. So how did those first cells make their living? Probably the most ancient cells from this point of view would be the Methanogens, which are archaea, so part of the archaeal domain, and the acetogens, which are bacteria. And they both live from the reaction of hydrogen and CO2, as you can see here. So methanogenesis produces methane as a waste product, and overall you can see it's exergonic, the delta G naught prime, minus 131 kilojoules per mole, so moderately exergonic, and slightly less so for acetogenesis. Whoops, I keep doing that. Where acetic acid is the waste product in that particular case. This acetal-CoA pathway is the only CO2 fixation pathway that's found in both bacteria and archaea. It's found in the methanogens and the acetogens. They're both growing from hydrogen and CO2. It's exergonic overall, and they're relatively simple pathways. So it looks ancient. That, of course, doesn't mean it is ancient, but it's quite simple looking chemistry, and it makes sense from a biological point of view. But you'll all know that hydrogen and CO2 do not react very easily together, and this has been a big problem in prebiotic chemistry over decades. Virtually the whole field has gone down a different line and looked at far more reactive molecules like cyanide and cyanoacetylene and so on, and used UV light as a way of energizing molecules and driving reactions. This has led to talk of terrestrial geothermal systems rather than deep sea hydrothermal vents, and the field is really very split. There is no consensus at all about the environment in which life started. So these views that I'm telling you this evening, I would describe them as not probably not the mainstream view. Neither are they a complete outsider's view. But they're probably not the mainstream view in chemistry. They're more the mainstream view perhaps in microbiology, because biochemists and microbiologists tend to think more about the actual pathways that you see in modern cells and wonder how they arose, whereas synthetic chemists tend to be more interested in what's the easiest way to make nucleotides at high yield in a one-pot synthesis. So there's different intellectual traditions at work here, and it's proved very difficult over decades to get hydrogen to react with CO2 to produce these intermediates. It's started happening over the last few years for reasons that I'll tell you. So what are these methanogens actually doing? Well, this is the geochemical pathway, and formaldehyde at the top here. So this is free energy landscape. So it's uphill to formaldehyde, and then it's downhill all the way through to methane. And most organic molecules are somewhere in this region here, so it's just slightly exergonic overall, but you've got to get past this energetic barrier before you're able to get there. And what methanogens do in particular is lower that barrier using a proton gradient. They are dependent on the proton motive force, on proton gradients across membranes, and we'll look into how that might work. So one question is, are there natural geochemical environments where there are proton gradients that could drive that reaction to allow it to happen? And the simple answer is yes. They were discovered again about 20 years ago now by Deborah Kelly, who was captain of the Alvin submersible. And this is Lost City vent field, which is in the mid-Atlantic, about 20 kilometers off the mid-Atlantic ridge. And you can see here that she's got a lump of a black smoker in her hand, and a bit of Lost City, and this looks like a sponge, more or less, a mineralized sponge. It doesn't have a chimney at all. The hydrothermal fluids percolate their way through these interconnected micropores. That kind of vent had been predicted to exist about a decade before they were first discovered in the ocean by Mike Russell, and his ideas through the 1990s had been considered, I think, pretty much out there. Not many people listened to what he had to say, but the discovery of Lost City changed all that. And this was Nature in 2006 did a feature article on him where they dressed him up as Erasmus, quite appropriate for this evening. And and called him, instead of a Renaissance man, they called him Naissance Man, as in the birth of life. And you can see here a bit of Lost City behind. all photoshopped up, and the reactor that he'd built at JPL at that time to try and simulate the conditions in these hydrothermal vents. And this at the right-hand side here is a cross-section of a part of the Lost City vent. You can see it's about 0.5 millimeter is this yellow scale bar down at the bottom. And so you can see that these are interconnected pores on quite a small scale. Lost City today, in an oxygenated ocean without much in the way of iron dissolved in the oceans, doesn't have anything like the kind of chemistry we anticipate that ancient vents in the Hadean period might have had, where there was no oxygen and probably a fair amount of ferrous iron dissolved in the ocean. So the anticipation is that these vent walls would have contained catalytic iron sulfide minerals. So not necessarily the entire wall. There would have no doubt been other minerals in there as well, but at least some presence of ferrous sulfides. High concentrations of hydrogen and CO2. These vents are produced by quite a different process to the black smoke events. It's basically a reaction between rock and water called serpentinization, and that produces strongly alkaline fluids around about pH 10 or 11, bubbling, almost literally bubbling with hydrogen gas. And so these are really electrochemical flow reactors and seem to be a very fitting environment for the origin of life. Everything I've talked about so far, rich in hydrogen. The early oceans there was a lot more CO2 dissolved in the early oceans. They were probably mildly acidic, perhaps pH 5 or 6. We don't really know exactly. With iron sulfide minerals in these barriers. So a lot of the conditions for early cells are met in this kind of an environment. And this was really what attracted me to this whole idea. My own background is in bioenergetics, and this is a cartoon of a bacterial cell, trying to pare it down to the absolute minimum basics here. The inside is relatively alkaline compared to the outside because there's a pump in the membrane, a thin phospholipid membrane, pumping protons out. So this is a kind of conceptualization of an autotrophic cell. Of course, everything's buffered, but this is an equivalent difference of about 3 pH units between the inside and the outside. And that's roughly what we have in a pore inside one of these alkaline hydrothermal vents. The outside is around about potentially pH 5 or 6, and the inside could be anything up to pH 11. Probably there's mixing in these systems, but you could imagine at least that there would be 2 or 3 or 4 pH units sometimes across these inorganic barriers. So is there any way, though, to go from these very simple systems to cells as we know them? And again, I turn to life itself to try to find a possible way, and this is what's going on in Methanogens. So this is getting slightly more complex, but it's really rather simple, so stay with me. This is a membrane protein called the energy-converting hydrogenase. It's using the proton gradient here, so it's a proton motive membrane protein to reduce ferredoxin. Ferredoxin is used to be known as the red protein. Red is reduced in this case, but it's containing iron sulfur clusters, 4Fe, 4S clusters usually, and ECH itself sitting in the membrane also contains 4Fe, 4S clusters, so iron sulfur clusters. So we've got iron sulfur proteins which are being modulated in some way by the proton motive force, which are using hydrogen to reduce ferredoxin. And the ferredoxin is then reducing CO2 through to a methyl group bound to a cofactor, and essentially that's going into carbon metabolism. So this is ATP synthase down here is necessary today. It definitely was not part of prebiotic chemistry. This is a sophisticated protein with a rotating nanomotor. So this is absolutely not prebiotic chemistry. So this must have come at a later date. There are other ways of making ATP, as I'll show you in a little while. So this is what I want to focus on, the energy converting hydrogenase here. And this is again a cartoon showing roughly what's going on and what the issue is here. So there are some membrane integral subunits which are a proton or sodium pore, and I think the proton pore is the most important one as far as the reduction of ferredoxin is concerned. We have several subunits here with 4Fe, 4S clusters and a nickel ion cluster here. So hydrogen is reducing the FeS clusters, the nickel ion cluster, and then these 4Fe 4S clusters, which are then reducing the ferredoxin. Now, under the kind of conditions that methanogens are growing, which is to say relatively low concentrations of hydrogen in the environment, the E prime is about minus 300 millivolts for hydrogen. That's assuming 10 pascals of hydrogen partial pressure. Whereas ferredoxin is 99% in the reduced state under normal cellular conditions, and the reduction potential then, the E prime, is about minus 500 millivolts. So hydrogen with an E prime of minus 300 millivolts has to reduce ferredoxin with an E prime of minus 500. So there's a deficiency of 200 millivolts, which is not to be sneezed at, and which is overcome by the proton gradient. So there's something about the proton gradient which is enabling this gap in reduction potential between hydrogen and ferredoxin. Now, the likelihood is all of this is depending on pH at the simplest possible level, at a prebiotic chemistry level. So the basic issue here is that hydrogen at pH 7, or indeed any other pH, is not reducing enough to reduce CO2 through to a formaldehyde. So we've got this gap here of close on 200 millivolts. It's about 150 to 200 millivolts difference, which is unable to reduce CO2. That's at pH 7 or at any equal pH. But if we switch through to the conditions in vents, so we put hydrogen gas at pH 11 and CO2 at pH 6, the reduction potential shifts. And the reasons for this are very simple. I probably don't need to explain it to you, but just in case, essentially, if hydrogen is passing its electrons onto something in an alkaline environment, the protons will react immediately with hydroxide ions, and so that reaction will be favored in an alkaline environment just by the Nernst equation, and so it's around about 59-60 millivolts per pH unit that hydrogen becomes more reducing. CO2, the other way around, it will pick up those electrons, but it's going to pick them up much more readily if there are protons available to balance the charges, and so again, the reaction depends on the concentration of protons according to the Nernst equation, and so it's again about minus 60 millivolts per pH unit. So in the conditions with pH 11 and pH 6, then hydrogen should comfortably reduce CO2. The issue here is that they can't mix, and this is why it becomes important that they are actually in a hydrothermal vent where you have some blocking of mixing because of this microporous structure. Now, I would have mentioned this last time I was talking. We built our first reactor about 10 years ago now. This was one of the early prototypes. We've had all kinds of problems with making these things work. We initially tried precipitating vent-like structures inside. We've had some success, but also quite a lot of failure. Eventually, we decided to switch to doing microfluidics. So this is a microfluidic chip with a relatively thick barrier that you can at least see inside. So we have laminar flow here inside the chip. You can see it a little more closely. And this thick barrier in the middle is a mixture of silicates and iron sulfide minerals. This is disordered McKinnowite that you can see here, which is an FeS mineral. And after a lot of trying, eventually a former student of mine succeeded last year. This was in PNAS last year. And he's done a rather beautiful job, though I have to say it's a rather limited job. But he has demonstrated conclusively, I would say, roughly what you're seeing at the left-hand side here. So this, I won't go through the NMR in detail, but just to show you what's going on. We've got hydrogen in alkaline solution, in hydrothermal solution on this side. The hydrogen's giving up its electrons to the barrier itself, leaving protons behind in the alkaline conditions that react with the hydroxide ions to form water. And across on the acid side, we've got CO2. It's picking up the electrons from the barrier and protons from the acid ocean waters percolating through, and it's picking up under these relatively acidic conditions. We have formic acid protonated. And what the NMR is showing is that the electrons really are, well, what it's showing is that the CO2, when using 13 CO2, that the formate really is coming from the CO2, which was added here. and it's showing using using deuterons that that the protons really are coming from the acid side and not from the hydrogen over here. This only worked when there was a barrier in the way. This only worked, in other words, mixing didn't allow it to happen. And it only worked at slightly high pressure. It didn't work at atmospheric pressure. It worked at 1.5 to 2 bars pressure. So it didn't require very much pressure, but just a little bit. And it didn't work if there were, so these down at the bottom here are showing where no signal at all. It didn't work if there wasn't a pH gradient. In other words, the pH difference is allowing hydrogen to pass its electrons onto the barrier, and those electrons are potentially crossing the barrier. It's possible protons are crossing the barrier the other way. We don't really know. But the protons definitely are coming from the acid side, and the CO2, the carbon in the formate is definitely coming from this CO2. So finally, that is demonstrated, only to make formate the very first step, but at least it's a concrete experimental demonstration now. But I've been worried about this for a while. Is it really electrons or protons that are crossing the barrier? And that was not fully demonstrated in that particular study. And this is, if on the right-hand side here, you could imagine a protocell sitting inside one of these pores. And to have a topological equivalence to an energy-converting hydrogenase in its simplest form, you have to have protons crossing the barrier and entering the cell and driving the reaction between CO2 and hydrogen. So this is the topology that having a modern cell would require at a protocell stage. And that would require that protons can cross this barrier, and presumably CO2 can cross this barrier as well. This reaction would probably be taking place right on the very edge of the barrier rather than inside the pore itself, but just for clarity, I'm showing it that way round. So for a true homology with the energy converting hydrogenase, it makes much more sense for protons to cross the barrier than hydroxide ions. And so we've had a look at that in our microfluidic chip, and this is showing that we do have laminar flow even without any kind of barrier at all. So we have the acid side, the alkaline side. It took us a while to find dyes that worked well enough, but you can see a small amount of mixing going on there. And then this is with a barrier as well towards the right-hand side. And it struck us immediately that this is quite a diffuse barrier with all kinds of aqueous inclusions inside the barrier. And you can see that these aqueous inclusions are all yellowy green color. In other words, they're mildly acidic. They're not alkaline. So we looked at that in more detail. You can see it much more clearly here using two different pH indicators. This is the alkaline flow channel, which is blue. This is pH 11. This is the diffuse barrier. This is the acidic flow on the other side. And you can see that the barrier, again, is acidic right up to the very edge here. And we measured the distances, and we were able to calculate. We stopped the flow, started the flow in the alkaline channel, the acid channel, calculated the rates at which protons crossed and hydroxide ions crossed. And eventually, it's clear that there's proton flux across this barrier is about 2 million-fold faster than hydroxide flux going the other way. And that means that there should be very steep pH gradients right at the alkaline side of the barrier. Steep meaning something in the order of 3 pH units across a single 25 nanometer McKinowite nanocrystal. So genuinely steep gradients there, not quite on a cellular scale, but getting very, very close to it. When I say cellular scale, I'm the membrane itself that these proteins sit in is only about 5 or 6 nanometers thick. So we're still practically an order of magnitude out in size, but we're getting closer. And so at least you can imagine, and this is again just to orientate you to some more experiments we've been doing, you can imagine that protons are crossing a barrier, that CO2 is being reduced on the alkaline side of the barrier, forming things like hydrocarbons or amphiphiles, which we anticipate would line the barrier, that they would interact with iron sulfide minerals and potentially enhance the reduction of CO2 through to organics. This CH2O is not supposed to be formaldehyde, it's supposed to just indicate organics generally. And so as we make more perhaps short, small polypeptides and so on, we would expect that rate of reaction to increase. And eventually we can imagine protocells forming perhaps even with proteins in the end, genes and proteins as a final stage. This I admit is pure imagination, but what's important about it is that it's showing there's a topological continuity all the way through from one stage to the next, and it makes a series of quite simple predictions that we can test and have been testing in the lab, and other people, of course, as well. So this is a paper from 20 years ago now from Tom McCollum and his colleagues, and what this is showing is that under hydrothermal conditions, it's possible to synthesize long chain fatty acids and fatty alcohols. So this is actually starting from formic acid rather than from CO2. This was done at slightly higher temperatures, 150 degrees and over, and at higher pressures. But conditions that you would expect to see in most hydrothermal vents. They were anticipating the gas water transition at those temperatures to release hydrogen and CO2. And what you can see here is chain length. So the chain length 10, 11, 12 carbons in alkanoic acids and alkanols. So these are the alkanoic acids, 9, 10, 11 carbons, and alkanols slightly longer. And this on the right-hand side is chain length for straight hydrocarbons. And again, there's a peak here around about 15 carbons in a chain length. So it seems to be feasible under these conditions to generate quite long chain hydrocarbons and amphiphiles, so long chain carboxylic acids and so on. These we wondered if these would form vesicles under the alkaline conditions, because there's been a question in the literature for a long time about whether vents are too harsh to form vesicles from fatty acids specifically, and they are harsh conditions. We're dealing with a pH range of between 5 and 12. We're dealing with temperature gradients of 50 to up to perhaps 100 degrees centigrade, at least modern ocean salinity, if not more saline, and then divalent cations like magnesium and calcium. These are the modern ocean levels of those, but there's no reason to assume that they would have been lower in the ancient oceans. And so if we can't make fatty acid vesicles that are capable of surviving under those conditions, then that's quite a strong negative aspect to this theory. Well, it turns out we can. We were absolutely delighted about this. So most of the literature has talked about decanoic acid. So it's used single amphiphiles. And what this is showing at the left-hand side here is that if you're at relatively acidic conditions below pH 7, then you tend to get globules of fat. Basically, the decanoic acid becomes protonated and will form globules. At more alkaline pH, you get micelles. But in this region here, which is rather steep with decanoic acid alone, you can get bilayer membranes forming. Now, if we mix 6 different fatty acids, and these are between C10 and C15 in length, you can see here that this area where you should be getting vesicles increases. So we're now going from around about pH 7 as before up to about pH 9, where you should be getting vesicles. And here, if we now have a 5-to-1 mixture of fatty acids and fatty alcohols, we're up to about pH 10. So this was a 5-to-1 mix. This was a 1-to-1 mix, and we lost this rather beautiful form of the graph and ended up with what looked like a horrible mess, and we weren't sure how to interpret this at all, except it suggests that there might be vesicles absolutely everywhere, and that's what you can see here. So this is pH 12, 12.13. This is the 1-to-1 mixture of fatty acids and fatty alcohols, and these are under modern ocean conditions. So 70 degrees centigrade, 600 millimolar sodium chloride, 50 millimolar magnesium chloride, 10 millimolar calcium chloride, and this is cryo-EM showing the protocells, and this is confocal microscopy, and again you can see rather beautiful cells under these really rather harsh conditions. So it plainly is possible to have vesicles with bilayer membranes forming under harsh alkaline conditions. Intriguingly, it's not so easy to have them forming below pH 7, because then everything becomes protonated and you no longer get vesicles. So it's much harder in acidic conditions than in alkaline conditions. And we were then wondering, well, can we trap iron sulfide minerals in these protocells? That was the original question, and we were really rather taken aback to discover when trying to do that actually we get immediately 4Fe, 4S clusters forming, just mixing with the amino acid cysteine alone at alkaline pH. So this only happened at pH 9. Once you'd formed them at pH 9, they were stable at other pHs as well. And at pH 9, because the thiol deprotonates with a pKa of around about 9, and so you get the negative charge on the sulfur, and that will then interact directly with iron. And remarkably, this is UV Vis spectroscopy. This is the spectrum. We looked particularly at this peak at 420 nanometers, and this is the concentrations of cysteine from 5 millimolar down to 0.1 millimolar, and so we just subtracted the baseline there. And we get quite a nice relationship with the cysteine concentration. We did some Mossbauer spectroscopy, which I'm not very good at interpreting this. Perhaps some of you will do better than I am. Again, it varies quite a lot with the cysteine concentration. This is 10 millimolar, 5 millimolar, 3.5 millimolar, and we get at 10 millimolar mostly. That's mostly mononuclear ion clusters. Whereas here we're seeing a mixture of different types of cluster: 4Fe, 4S clusters, 2Fe, 2S clusters, and mononuclear ion clusters. And we were able to show that these are redox active as well, not as reducing as I would like them to be. This was a silver electrode, so this is minus 400 millivolts on a silver electrode, which is about minus 200 millivolts on a standard hydrogen electrode. So they're moderately reducing, but it's remarkable that simply mixing a single amino acid in solution at pH 9 produces 4Fe, 4S clusters straight away. We've been trying to get them to interact with the membrane, but we've not had a great deal of success with that yet. We've taken this a little bit further by thinking in modeling terms, what would we expect to see, and where will that lead us in terms of experiments? So this was our original conception in the model, that we would have an interaction between amino acids and iron sulfur clusters to give something equivalent to an energy converting hydrogenase, and that's roughly what we've managed to do with the iron sulfur clusters and the cysteine, but we've not managed to get it into the membrane yet. We think that should reduce CO2 through to at least C2 intermediate, so something equivalent to acetal-CoA, perhaps thioacetate or something. And this would branch to give fatty acids or amino acids. And so the question is, can we have? We would expect catalytic feedbacks from amino acids on CO2 fixation, and that perhaps with a greater flow through would lead to more branch points. So we're anticipating the branch points of modern metabolism. A lot of this ought to work, and some of it has been demonstrated already, but some by us, some by other people. So we can form amino acids from Krebs cycle intermediates. That's relatively simple. Sugars have been done by Marcus Ralzer and colleagues, starting from phosphoenol pyruvate. So difficult to know exactly where the phosphoenol pyruvate's coming from, but from there you can get through to ribose and glucose. An energy currency acetal phosphate, we've succeeded in making that. And fatty acids, I showed you the Tom McCollum experiment, so not exactly under the same conditions, but rather similar conditions. These are the, as I showed you at the very outset, these are the precursors for nucleotide synthesis. So if you're able to make these at sufficiently high concentration, we ought to be able to make nucleotides. I have a PhD student who's been working on this, and he's actually done most of the steps now from pyrimidine synthesis, but there's still one or two which are missing. And we then anticipate that nucleotides would feed back on CO2 fixation, as they still do today with folates, for example. And we can imagine all kinds of other feedbacks as well from nucleotide catalysis, and we can test those in the model to see what would we anticipate works, what doesn't work, how would we expect it to be. If you're diverting all this flux down different pathways, then there would be less going to fatty acids, less going to amino acids, and so you should have less flux than in this first place. But if you have a feedback from nucleotides catalyzing CO2 fixation, relatively strong rates of catalysis, you begin to take off. You have an exponential lift off in the rates of growth. I won't go through this in any great detail, but we were able to show that you would expect to see with just nucleotide catalysis, you would expect to see an increase in the rate of protocell division. What we were hoping for would be also a concentration of nucleotides inside the cells, and we don't see that. We're able to concentrate nucleotides in silico if we have this autocatalytic loop feedback here. But in a protocell, in the context of protocell growth, it would actually slow down protocell growth. And so if selection is on the growth of these protocells, you're never going to accumulate nucleotides inside, which is an interesting conclusion to come to. So exactly how polymerization is taking place is a difficult question for us at the moment. I just want to finish in the last 5 minutes or so with some more experimental work, a couple of beautiful bits and pieces here. So this is looking at the formation of, the phosphorylation of ADP to form ATP using acetal phosphate. And this is work from Silvana Pinner. And what you can see here is that at pH 5.5 to 6, or at pH 7 and 9, so much less effective at alkaline pH in this case, this is the yield of ATP from ADP and acetal phosphate. So 15% yield roughly. And this is from carbamoyl phosphate, and then down here are a whole bunch of other things which didn't work at all, but you can see phosphate alone, cyclic trimetaphosphates, phosphoenol pyruvate. This is pyrophosphate, pyrophosphite, carbamoyl phosphate, and acetal phosphate. So we tried quite a range of phosphorylating agents, and really acetal phosphate works well at a slightly mildly acidic pH. Carbamoyl phosphate is the only thing that works at all well otherwise, and acetal phosphate doesn't work very well at pH 9. We also found, very interestingly, that acetal phosphate will phosphorylate ADP to ATP, and you can see the peak here after 3 hours. But if we try with IDP or GDP or CDP or UDP, we don't get any peak at all. This is the control, so and there is no increase over the control values of any of these. And so it does look as if there has to, this amino group on adenosine is absolutely critical for this reaction to happen. And it seems as if it's critical that acetal phosphate rather than other phosphorylating agents is required for phosphorylating ADP to form ATP. So nothing else works. So the fact that ATP is still the universal energy currency in biology seems to go all the way back to prebiotic chemistry, to the very early conditions. Acetal phosphate, I should say, is still used in substrate level phosphorylation. It's the bridge really between thioester chemistry, so acetal CoA and so on, and phosphate chemistry, ATP. So acetal phosphate is the bridge between these two worlds. It's still used today in bacteria and archaea, and apparently in eukaryotes as well. It's formed transiently in pyruvate dehydrogenase, and a lot of this seems to have been defined very early on in prebiotic chemistry. Oh, one other thing I should have said, it only works in the presence of ferric iron. I have no idea why that is. That's something different. We also tried in the presence of a panel of about 20 different ions, and ferric iron is the only one that worked. Ferrous iron, for example, doesn't work. It seems to be the charge density of ferric iron which allows that to happen. Okay, I'm going to, this will be the final slide, I think. We're thinking about the code itself. How does the genetic code originate in this context, in the context of growing protocells? And there are various patterns in the code that you may or may not be familiar with, which relate to hydrophobicity. And so this is a pattern between the hydrophobicity of the first 2 bases in the anticodon, so this is the hydrophobicity rank, against the hydrophobicity of the amino acid itself. And you can see it's a reasonable relationship there. The size of these balls relates to the antiquity of the amino acid that it's coding for. So the larger ones are the more ancient ones here. So valine, for example, aspartate. Have I got this the right way around? Sorry, this would be, no, I've got that the wrong way around. I beg your pardon. So the small ones are the most ancient. So this is valine. This is aspartate here. So these are some of the more threonine. Alanine is quite an ancient one. And where's glycine gone? Can't find glycine. Glycine is here. So these are among the more ancient. So we see quite a strong relationship. All we need really to explain the origin of the genetic code in the context of these protocells is to assume that the polymerization will take place within these protocells, and that there are interactions between specific amino acids and anticodons. And this is something which people have tested over some time. We are trying to do it. There are some suggestions in the literature that there are weak but consistent interactions between anticodons and their cognate amino acids. We're looking now at NMR. I don't really have anything to show you yet on that, but we're beginning to get some signs that there really are some weak but consistent interactions between the anticodon and the cognate amino acid. And that then it basically solves the issue of where does information come from in biology, because it's really biophysical interactions in a particular setting, which is coming back to what I was saying at the beginning, the growth of protocells. These protocells are capable in principle of growing, of doubling themselves. We've seen that nucleotides potentially can form within them. We've been attempting nucleotide synthesis, and we've done a lot of the steps. It's very difficult to put all these things together. But the thought is then that nucleotides will be formed inside these autotrophically growing protocells. That the nucleotides eventually, for reasons that we don't know exactly why yet, will polymerize to form short random sequences of RNA within these protocells, which will interact with amino acids for biophysical reasons, and can generate functionality then at the level of, is it a hydrophobic protein or polypeptide, or is it a hydrophilic one? And where does it go? Does it go to the membrane, or does it go to the cytosol? And its function, to some extent, will depend on its position within the protocell. And so we're able to answer, at least in principle, where information is coming from in biology. So how does chemistry come alive? That was my question. And here's a go at answering it in a single slide. It happens when a focused, sustained environmental disequilibrium of hydrogen, CO2, and pH across a porous structure that lowers kinetic barriers to reaction continually forms organics that bind and self-organize into protocells, with protometabolism generating catalytic nucleotides which promote protocell growth through positive feedbacks, favoring physical interactions with amino acids, which is this nascent genetic code that I've talked about, where RNA sequences are selected if they promote protocell growth, so genetic information has meaning from the beginning. That's my best shot at explaining how chemistry can come alive. And that's it. I just want to say thank you, especially to my students who have been very brave, I would say, in working on this. This is the largest my lab has ever been, in fact, at the moment, and we are making some progress. You can see that they're holding the Krebs cycle there. I hope you recognize that. And thank you for listening. Let me stop sharing and answer some questions.
Thank you very much indeed, Nick. That was
my pleasure.
Let's look around and see if we have people flagging that they have questions. Not at the moment.
No.
Let me throw a general spanner in the works. One of our old Imperial College mates, who may have been around in your days as well, Brian Hartley, used to say that the problem with evolution is that we weren't there when it happened.
Mm-hmm.
Is this still one of the major problems? A lot of it's very hypothetical.
Yes, of course it is, and we'll never know what happened because even if we could build a time machine and go back, we wouldn't know what to look for. We wouldn't know what was leading to life, what was on the path, or what wasn't. Even with human evolution, we're not really sure which hominoids were on the path to humans. So, no, you're quite right, or he was quite right. We'll never know. What we can do, though, I think, is try to lay out, in principle, what all the steps are to go from a purely inorganic world to cells as we know them, and to test each and every step along the way. Ideally, we ought to be able to put them together But of course, it was a whole planet that was doing this over an unknown number of millions of years. So the chances of us being able to do that, I would say, are remote. But the chances of us being able to show that it's possible. I mean, next steps for me would be to show that we can put these iron sulfur clusters in a membrane, and that in the presence of a natural proton gradient, they will reduce CO2 to make more organics. For me, that would be a big step. For probably anyone in the audience, that might be almost laughably trivial because it's already been done, but not in that setting, not in that way, not as cells do it in the simplest prebiotic way. So I don't think we're wasting our time. I think we can understand in principle how it works, but I agree with you that we'll never know what actually happened or if life really started this way.
All right. Tim?
Oh, hi. Hello. Yeah, a question that comes to mind is, I mean, in doing this research, has your results make you think that there's a possibility of life elsewhere in this solar system is possible? I mean, because I can imagine that the ingredients that you're talking about there, I mean, in terms of pH, hydrogen and carbon dioxide, could be found
very common, yes,
anywhere really. I mean, given
well,
the right sort of,
I mean, the most interesting possibility at the moment would be Enceladus, where there are plumes coming from the surface through cracks in the ice on the surface of Enceladus, which go out, I think, about 200 miles from the surface of the moon. So one of the moons of Saturn. Yeah. And it's alkaline, pH 10, I think, if I remember rightly. There are organics dissolved in there, and there's hydrogen in there as well. So this was from the Cassini flyby from spectroscopy on that. And so, yes, the same conditions would appear to be true of the ocean underneath the ice on Enceladus itself. Likely you'd see similar things on Europa. And there is plenty of evidence of serpentinization having happened on Mars in the past, and plenty of water on Mars in the past as well. So I'd say it's doubtful that there's life on Mars now. Maybe there is beneath the surface somewhere, but there's not much sign of it anyway, in terms of planetary atmospheres. But it would be very disappointing if we didn't see something on Enceladus.
Yeah, look forward to it.
Another question? Yeah. I was intrigued by one observation you made, Nick, that you would think that the levels of iron sulfide deposits in these hydrothermal vents was much higher earlier in evolution than it is now. What's the reason for thinking that?
Well, there are plenty of banded iron formations dating back 3.5 billion years or more, implying that the early oceans were ferruginous. Right. Whether that's really true is coming under some scrutiny now, because it's possible that it was in fact coming out of hydrothermal systems, and was being oxidized later on. So it's not really clear what exactly the concentration of ferrous iron in the early oceans actually was. Most people would say low micromolar quantities. Yeah. So not huge amounts, but enough for the kind of chemistry I've been talking about. Whereas now there's essentially none. Yeah, and we know that seeding large amounts of iron into the oceans creates big algal blooms, doesn't it? Absolutely, it does. Yes. Yeah. So I mean, this is the trouble. As soon as you have oxygen, iron will precipitate out as rust immediately. So iron availability was much greater before oxygen levels rose, and again, it's disputed when oxygen levels rose, but basically after the great oxidation event around about 2.3 billion years ago. So the first 2 billion years of life on Earth were, if not anoxic, very close to it. Very close to it, yeah. Which greatly favoured ferrous iron over ferrite. Absolutely, yes. Yeah. And also it favoured organic synthesis, because as soon as you've got oxygen around, it's going to react with any hydrogen much faster than CO2 is going to react with that hydrogen. And so to favour the kind of organic chemistry I've talked about, you require pretty strict anoxic conditions. It really doesn't happen even at low levels, trace levels of oxygen. David, please.
Is it possible to apply the knowledge that you've gained to solve the problem of global warming?
Very interesting question, and in principle, yes. In practice, I suspect it will be very difficult. But essentially, what we're talking about is a way of using pH to drive the reaction between CO2 and hydrogen. So it would strip CO2 from the atmosphere and generate hydrocarbons, which could be then burnt as petroleum or used in the chemical industry. So and it would be net zero carbon emissions. So it would, in principle, work extremely well. In practice, how easy it is to do it. I mean, at the moment, it requires high pressure. That would probably be economically difficult. But if we can find a way of making it work to our own satisfaction, I would love to talk to chemical engineers at that point and see if we're able to come up with a way of making it work in an economic way. It should work. Mm-hmm. I mean, an issue to me at the moment is it electrons crossing the barrier or is it protons crossing the barrier? Because if it's electrons, then really it ought to be quite simple. All you really need to do is put a copper barrier there. So I'm going to get a student to look at that fairly shortly, just to nail that one at least.
It's intriguing, isn't it, that in fact we again come back to the situation where we're looking at the importance of pH gradients as a driving force. Because
I mean, it seems almost comically simple, doesn't it?
Force for ATP synthesis in mitochondria.
Yes. I mean, this is how I came to the whole question. Really, was why on earth does bioenergetics work this way with proton gradients across membranes? And I think for a long time I got stuck with the idea that it was about ATP synthesis, and it's virtually impossible to think of any prebiotic intermediate between an ATP synthase and a simpler form of it. I mean, it's I just can't think of anything. Yeah. Whereas CO2 fixation, driven by something like the energy converting hydrogenase, it's still a proton motive membrane protein, but it's a much simpler one, and you can then imagine at least prebiotic intermediates, which are simple iron sulfur clusters collated by either single amino acids or more likely short polypeptides. Yeah. And again, an interesting question that I have a student working on now is what is the simplest possible proton pore in a membrane, in a simple membrane? I mean, is it really just a single helix crossing the membrane? Would there be enough trapped water in that to allow protons to cross relatively rapidly? It's amazing what work hasn't been done. You know, these are simple questions that shouldn't be very difficult to answer. Will take us a while because no one's ever really done it before, and so we face our own problems as we go along.
Have we other questions? No. Well, in that case, let me take this opportunity again for thanking you for a brilliant lecture. We've enjoyed it very much. Look forward to having you again with us next time in the flesh, and I promise you will get a meal instead of just a little gesture of our appreciation, which arrived, I'm glad to hear, this morning. So
thank you very much for that.
Not at all. Just to remind our members and friends that the next activities we're coming up, we have John Donoghue from Trinity College Dublin, who's going to be talking in September about chemistry and the cinema. We've got Pierre de Klerk, who's going to give us a talk on Kekulé in Ghent and lead us to where the RSC plaque that we placed there, the blue plaque that we placed a few years ago in the chemistry department, has now migrated into the centre of Ghent, close to where he had his idea of the cyclic structure of benzene. Philip Ball in October on the beauty of chemistry, and then a talk on battery technology from Jean-Francois Gouy in November, the Café Chimique in January or February, and the RSC president, Tom Welton, on green chemistry, sustainable chemistry in February. So there's plenty of events coming up, but we'll keep you posted on our website. Thank you all for joining us once again. Have a good weekend, good time until next we get together again in September, hopefully in person, not just in virtual form. And thanks once again, Nick, for your contribution. Enjoyed it very much.
My pleasure. Thank you.
Thank you.
Bye now.
Voilà.